Hello, everyone. Just give everybody a moment to enter the room here. All right. I'm going to go ahead and get started just so we have enough time to get through everything. So hello again, everyone. My name is Brianna Montgomery, and I would like to welcome you to the Autism Intervention Research Network on Physical Health, the ARRP June webinar. Thank you all for joining us today. Because of the number of participants, your audio will be muted throughout the call. However, you can submit questions at any point during the presentation via the Q&A feature or in the chat box on your webinar console. Just a friendly reminder to please be respectful when communicating in the chat and asking questions, and note that only questions or comments pertaining to the presentation will be addressed. This entire webinar is being recorded and will be available on the ARP website at arpnetwork.ucla.edu. I will also place this link in the chat. You may also view the presenter slides and follow along at our Google Drive archive that will also be placed in the chat momentarily. There will be a short evaluation survey at the close of the webinar. We invite you to provide feedback on this webinar and also to provide suggestions for future webinars. In the interest of time, let's get started. We first want to acknowledge the HRSA, the Health Resources and Services Administration, as the funding source for the AIRP. It is my honor to introduce our presenter for today, Dr. Christine Wu Nordahl. Dr. Nordahl is a professor in the Department of Psychiatry and Behavioral Sciences at the UC Davis Mind Institute. Her research program utilizes structural and functional MRI scans to study brain development in autism across the lifespan. And since 2006, she has led neuroimaging efforts of the Autism Phenome Project and Girls with Autism, Imaging of Neural Development Study, two integrated longitudinal interdisciplinary studies aimed at identifying clinically meaningful subtypes of autism. Please join me in welcoming Dr. Nordahl.
Thank you so much, Brianna. I'm thrilled to be here, and let me go ahead and start sharing my screen. All right, does that look okay? Yep, all right. So I'm gonna go ahead and get started then. Just to let everybody know the plan, the topic today is autism in girls and women. I'm planning on presenting for maybe 45 minutes or so. I'm gonna try to leave plenty of time for questions. Love to hear from you. I think we're going to try to save questions until the end, but if there are any quick clarification questions, or if I just say something that's not clear, feel free to drop them in the chat and I can try to answer those in real time. So let's see. So this is again to acknowledge HRSA and the AIRP grant. I'm really grateful to have this opportunity to speak with you today. So the objectives of the presentation today is really to go over some of the current theories about why there are fewer females diagnosed with autism. I'm going to go over some of the current autism literature on what we know about behavioral sex differences in autism, and go into a little bit more depth on the current literature on masking and camouflaging in autism. Because that's a topic of great interest and I think clinical relevance. And then finally, I will go over some recent findings from our UC Davis Mind Institute GAIN study for girls with autism, imaging of neurodevelopment. And there I'll go into some of our brain and neuroimaging findings to tell you a little bit about what we've been doing for the past few years in this study. First, I want to just make it clear that throughout this presentation, I will be referring to female as a biological sex. Everything I'm talking, all of the studies I'll be talking about here are utilizing biological sex assigned at birth to evaluate differences between males and females, recognizing that there are definitely going to be gender differences as well, but that's not something that I'm going to be able to focus on today. So I just want to provide that disclaimer at the very beginning. So one of the most consistent findings in autism research, if not the most consistent finding, is that there is a skewed ratio of boys to girls as far as the number of children diagnosed with autism in the US. This figure is just showing that when I got into autism, the field of autism back in 2000, the prevalence rate of autism was 1 in 150. 2016, it's 1 in 54. But the ratio of the number of boys diagnosed with autism to girls has remained fairly constant at about 4 to 1. So 4 boys diagnosed for every 1 girl. And what's become really apparent over time is that the majority of research study samples reflect this bias. And so autistic females are relatively understudied, but I think in the past 5 or 6 years, this has really started to change. So there is hope there. And notice that I'm talking about 4 boys diagnosed for every girl. I think it's important that we're still working out what this exact ratio is, and whether it's a diagnostic issue or whether it's children who actually have autism. And I'll get into that a little bit more. So the $1 million question then that we've been working on for a little while is why? Why are there fewer girls with autism? And there are really 2 prevailing theories out there. And we've made some headway in exploring both of these theories. They're not exclusive of each other. I think there are just 2 different explanations as to why there are fewer girls diagnosed with autism. So the first theory is that girls are underdiagnosed. This is a behavioral theory suggesting that somehow girls are flying under the radar, and that prevalence ratio of 4 to 1 isn't actually as drastic as we think. There have been population-based studies suggesting that skew in sex ratio is actually much less than 4 to 1, that it's maybe 3 to 1 or 2 to 1. And so we're still working out those exact numbers. But I think that certainly there is evidence now that I will present to you today that girls are going under or misdiagnosed. But I don't think that explains all of the skew in the sex ratio. I think that there are still biological differences that are leading to fewer girls having autism. And the main theory that we've been working on as a research field to describe why there are fewer girls with autism from a biological perspective is this female protective effect. And I will tell you more about that as well. This female protective effect really comes from genetic studies. And I think, so it's my next slide. So I will go into it a little bit more here. This is a term you maybe have heard of before. For me, it was something that was confusing. You see this curve and you hear female protective effect, and what does it really mean? So I just want to take a little bit of time to walk you through what this theory is and what we mean when we talk about the female protective effect. So on this graph, we're looking at the population. The blue curve is we can think of as males in the population, the red curve as females in the population. And what's important here is that there's this liability threshold. And we're thinking about this in terms of a liability threshold for a diagnosis of autism. So some alternative names for this model are called the differential liability model or the multifactorial liability model. Those are 2 alternate names that you'll also hear the female protective effect described as. And what it suggests is that there's some sort of etiologic load. So this is genetic load or environmental load, something that if you have an increase in this load, it moves you closer to this threshold of diagnosis. And that there may be female-specific factors that push you further away from this diagnostic threshold. So this is all still very sort of, for me, difficult to digest. And so I actually asked my daughter years ago, in trying to explain it to my 10-year-old daughter years ago, she actually came up with some illustrations for me. So let me walk you through it. The idea here is that you have a person. And here I'm just going to think about these rocks as this etiologic load. So we can think about it in terms of genetic hits or environmental hits or a combination of genetic environmental hits. And all of us can go around carrying this load and not having a diagnosis of autism until that burden becomes too much. When you have too many genetic hits or too many gene environment interactions, that load becomes too much, and you've crossed this threshold into a diagnosis of autism. So this is illustrated by the boy then not being able to carry this load anymore and dropping his pile of rocks. Now, when we think about the female protective effect, we think about something specific that females have that allow them to have more of this load. And so in this illustration, this analogy, you can think of the female protective effect as this girl actually has baskets. And so she can carry more of these genetic hits, more of this etiologic load before the burden becomes too much and she crosses that threshold into autism diagnosis. So this is sort of a simplistic way of thinking about this model of the female protective effect. Of course, what we're trying to understand is what are those baskets in terms of biology? You know, what is the protection that females are gaining? Or on the flip side, what is making it so that males have higher likelihood of having susceptibility to some of these hits? And so this is just a quick run through of what we mean by the female protective effect. And just very quickly, I think the most straightforward evidence and direct evidence that we have for it have come from genetic studies that have shown that autistic females, when compared to autistic males, actually do have more frequent and more extensive copy number variations. So this is a type of genetic mutation than do autistic males, suggesting that that there is evidence supporting this female protective effect. Now, I'm a neuroscientist, and so what I think that I've been studying is how do these differences in genetics then translate over to the brain? Is there a different neurobiology in males and females with autism? Even if the behavioral outcome is the same, there could be different neural pathways leading to that outcome, and that's what we're trying to understand in my lab. So hopefully that made sense. That's a quick overview of the biological theory. Now I want to go back to this idea of exploring what the evidence is for females being mis- or underdiagnosed with autism. And here I just like to go through this study. It's 10 years old at this point, but I think it's still one of my favorite studies, and I think it illustrates this point really well. Let's see. So this was a study done out in the population with 15,000 twin pairs, and they're basically surveying the population in terms of giving a parent a report of rating their child's autistic traits, and all the children were 8 years of age at the time. And they split up the children into two groups. They split up a group of children who had a very high autism traits, but no clinical diagnosis of autism. And then another group who had high autism traits plus a clinical diagnosis of autism. And then they simply looked at the biological sex breakdown across these two groups. And what they found was that there were fewer girls actually with high autism traits who also had a clinical diagnosis, if that was all that they had. So girls who had a clinical diagnosis based on their high autism traits also had higher levels of intellectual disability or other behavioral challenges, suggesting that girls who had high autism traits alone without these other sorts of behavioral issues or cognitive issues were not going in and getting a clinical diagnosis, suggesting that these girls were flying under the radar, if that makes any sense. So girls were less likely to receive a diagnosis of autism despite having autism traits unless there were additional problems. And I think that this was a really nice study to really illustrate 10 years ago that there was this problem of girls going under or misdiagnosed. So another question then would be, why then? Why are girls flying under the radar of diagnostic radar for autism. And I think a really fruitful area of research, and I think something that we've really learned from the autistic community, is that autistic females may be more likely to try to mask their autism symptoms. And so this masking is one way that people have described it. Camouflaging is another term that people have used. Here I'm just going to lay out some definitions. Social camouflaging being the use of strategies by autistic people to minimize the visibility of their autism during social situations. And I think that one of the most famous autistic people is Temple Grandin, and she gave this great quote: "The thing about being autistic is that you gradually get less and less autistic because you keep learning. You keep learning how to behave. It's like being in a play." And so I think that what she's describing here is masking her autism symptoms, trying to minimize the visibility of autism characteristics when you're interacting with non-autistic individuals. And for her, it was like a script. It was like being in a play. Another quote that I really like to bring up is from Dr. Summer Bishop, who's a clinical psychologist at UCSF. And this quote is an excerpt from a Spectrum News article that she wrote several years ago. And I thought this was, I think this situation has been played out in the Mind Institute clinic as well. So let me just read through this. "One 6-year-old girl I met several years ago seemed at first to have good social skills. She responded appropriately when I introduced myself, complimented my outfit, and politely answered all of my questions. It was only when I saw her again a few days later that I understood her family's concerns. She made nearly identical overtures as if our interaction were part of a play she had rehearsed." And so what I always think about here is that if Dr. Bishop, who is a brilliant psychologist, if she had only seen this girl once, then she may not have diagnosed her with autism. It wasn't apparent until she saw her again that this little girl was essentially masking her autism characteristics to fit in with how she thought she should be fitting in. And I think that this is maybe a scenario that's been played out in many diagnostic assessments of little girls with autism, that they can sort of fake it for a little while, but you really need to keep probing to draw out the real behavioral and social challenges that she may be experiencing. So again, this is, I'm just sort of showcasing some of the research that's been done on camouflaging and masking. And there are some really great studies out there looking into what camouflaging is, how you can score it, how you can rate it, both from a parent and a self-report perspective. I like to highlight this study, the playground study, because it's an observational study of camouflage. And I just think that it's a really great design and really interesting findings. And so in this study, the observers were just watching children, second grade children, during recess. And they were coding the types of play behavior in 1-minute intervals that these children were participating in. And the types of activities, the groups of activities that they were coding were games. So structured games like handball or tetherball or foursquare, always a favorite on the playground. They also coded behaviors that looked like solitary behaviors. So when children were alone and not engaging with others. And then they also looked at joint engaged behaviors. So talking and active socialization in groups. And then I think what was really interesting is that they also looked at this behavior called flitting. So how often did a child go between these different types of activities during their recess? And what they found, so here I'm just going to break down this table for you. So we have typically developing boys and girls, as well as boys and girls diagnosed with autism. And the time, just simply the number of seconds in that 1-minute interval that they spent in games, joint engage, or solitary. And so the first thing that's perhaps not unexpected if you've been around second grade boys and girls is that the non-autistic or typically developing boys spent most of their time in games, playing soccer or structure sort of games. Strikingly, the autistic boys spent most of their time in solitary as opposed to games. And this is not a surprising finding. This is what they would have expected. But what they found that was interesting was that both typically developing and autistic girls spent most of their time in joint engaged sort of behavior. So even though the autistic girls were spending less time than the typically developing girls in joint engaged type behavior, they were still spending most of their time, so 39 seconds as opposed to 6 and 26 seconds in solitary. So they were spending most of their time in joint engaged. Autistic girls were also spending a fair amount of time in solitary play as well. What they also noticed was that TD girls would flit from joint engaged to joint engaged. So they would flit from social group to social group, sort of moving from social group to social group and fitting in. Whereas autistic girls did not have as much of this flitting behavior. They were not really engaged with these groups. Let me say this again. So the autistic girls were flitting between the joint engaged and solitary behavior as opposed to flitting between the joint engaged and joint engaged. So flitting between social groups and flitting just between a group and themselves. And what the authors of this paper point out was that although autistic girls knew that they should be within these social groups and were sort of hanging around in these social groups, they were never really fully immersed in these social groups. And so if you were looking at this group of kids just from the outside, if you were looking as a playground attendant, you may miss the autistic girls because they looked like they were in these groups, but their social challenges were apparent to the girls that they were interacting with, and therefore they weren't really fully immersed in these groups. And I think the take-home message of this study was that these girls were masking their behaviors, masking what it was like for them to be autistic on the playground. And if the practitioners were only looking for social isolation on the playground when identifying children with social challenges, then autistic girls would continue to be left unidentified because it would look like they were parts of social groups. So I think that this is, a really nice real world example of what it can look like for an autistic girl on the playground and how they could potentially be missed. So now moving on to what we know in other areas of research that have come out in the past. These studies have all come out in the past 2 years, all pretty strikingly consistent that the age of diagnosis for autism is later, about 1.5 years later in girls than in boys. And so across different networks, very large studies in Rhode Island, in Canada, in Uganda, and across the US. In every study, girls were diagnosed later, and alarmingly, girls were usually diagnosed around 7 years of age as opposed to about 5 years of age in boys. And what this means is that girls are really missing out on getting critical intervention or supports that they could really be benefiting from. So I think this is a cause for alarm in trying to get girls in for more early and accurate autism diagnoses. Some other things that we know about differences in core autism characteristics that have come from several studies now, looking at behaviors in children. Several studies report that boys have more severe restricted and repetitive behaviors than girls. This is a little bit in question because there is evidence that the diagnostic assessments that we use have been designed primarily in boys with autism, and that when we think about restrictive and repetitive behaviors, we really have to think about gender norms as well. So girls may not be as interested in things like train schedules, sort of the stereotyped what you would think of in terms of restricted and repetitive behaviors. Maybe the girls have restricted interest in purses or lunch boxes or My Little Pony, which could seem a little bit more gender appropriate and therefore missed. There have been also some studies looking at social communication differences across boys and girls. What studies have generally found is that there are fewer differences in early childhood, or girls seem to have better social communication skills in early childhood. But looking longitudinally as these girls grow up, they seem, based at least on parent report, to be having more severe problems during adolescence. And again, I think that there are gender issues here going on, whereas earlier on, the social dynamics of being in a play group or something like that are a little bit less complex. If anybody has any teenage girls at home, things get very challenging in adolescence, and maybe harder for autistic girls to be masking their behaviors as they get older. And so I think this is another area where we really have to look into supports for autistic girls, based on finding these challenges they're having. This is a sort of a call for finding better supports and accommodations for autistic girls as they get older. And so I just basically talked about this trajectory of autism symptom characteristics, that girls seem to improve across early childhood, but this could be related to just the social dynamics as you get older, become more complicated in adolescence. And so this pattern might reverse in adolescence. What do we know about some of other behavioral differences between boys and girls diagnosed with autism? So again, here the key is diagnosed with autism, because that's what we've mainly been studying in the field. There's a higher proportion of diagnosed girls who have intellectual disability, 39 versus 32%. So that's going back to the first study I talked about, that maybe girls are not even brought in for a clinical assessment unless they have other co-occurring problems like intellectual disability or psychiatric symptoms, and that's getting them in for a diagnosis in the first place. It's really likely that girls without intellectual disability are going underdiagnosed. What we've also been focusing on a lot is looking at co-occurring psychiatric conditions and other developmental conditions in girls with autism. And there have been studies showing that a higher proportion of girls diagnosed with autism have co-occurring symptoms of anxiety and depression, even as early as age 3. And I'm going to be going into more detail about one of our studies that has shown that really in girls as young as 3 years of age. There have also been studies showing that females have higher rates of co-occurring conditions beyond psychiatric symptoms, so developmental, psychiatric, and neurologic conditions as well. This could all still be going back to this idea, though, that of this diagnostic issue of maybe the girls who are getting diagnosed with autism already have these higher rates of co-occurring conditions, and there are girls with autistic traits only who are going underdiagnosed or flying under the radar. I think one really striking finding that we as a field and as a community and as people need to pay attention to is that autistic women, we know, are at higher risk than autistic males for attempted suicide. And there's some research suggesting that the higher, this masking that I've been talking about, this camouflaging, it may work well in social situations, but it doesn't come without a cost. And I think that there is some evidence that people who camouflage or mask more tend to be women, and people who mask more also have higher rates of attempted suicide. And so I think we really, really need to think about what kind of supports and wraparound services we should be providing to autistic women, or at least be on the lookout for in terms of what we need to be doing to help them. So now let me turn to my study, the GAIN study, and let me tell you a little bit about the history of this GAIN study in the context of the Autism Phenome Project. And so we started this study back in 2006. When we started the Autism Phenome Project, our goal was to try to identify children as young as possible, right as they were getting diagnosed, so around 2 to 3 years of age. And we wanted to take a very comprehensive look at all different aspects of their behavior and medical and immune profiles, so that we could try to identify what we think are clinically meaningful subtypes of autism. So the premise of this study comes from the idea that we know that autism is very heterogeneous, and there's likely multiple subtypes of autism. And as researchers, when we put all of these subtypes together and compare them to non-autistic controls, we're maybe losing a lot of individual differences between these subtypes and losing a lot of information when we just group them together. So we're trying to identify subtypes perhaps that have certain immune conditions, or subtypes that have gastrointestinal issues, a subtype that has intellectual disability, a subtype that maybe has co-occurring anxiety, and then try to understand the causes and the treatments and interventions and supports that are most meaningful and relevant to each of those subtypes. So that's the goal of the Autism Phenome Project. But like all the other studies going on, when we recruited our cohort of children, we had about 300 families involved, but we had many, many more males than females. Autistic males were recruited at this 4-to-1 ratio of females. So we had about 200 autistic boys in our study and less than 50 autistic girls in the study. So sorry, this is mislabeled, or this is unlabeled. These columns are the number of autistic participants. This is the number of our non-autistic controls. So seeing that we had this imbalance in the sex ratio of autism, we initiated the Girls with Autism Imaging of Neurodevelopment study, or GAIN study. It was funded in 2014 to try to increase the representation of autistic girls in the APP cohort so that we could begin to assess sex differences. And so what you can see is that we were able to triple the number of girls in our study. We now have over 100 autistic girls in our study. We still have more boys, but that skew and ratio at least has gone down a little bit. Importantly, we have almost 100 girls with MRI scans in our study, and that is more than enough to start looking for neurobiological sex differences. And we also have comparably numbered non-autistic control groups now. So we have about 100 or about 80 non-autistic females and 80 non-autistic males that we can use as our control groups in our study. Our study design is that we enroll children at 2 to 3.5 years of age, and we collect a lot of information on them. So we collect MRI, a lot of behavioral assessments and cognitive assessments, medical information. So we do a med exam and look at family history and GI symptoms and immune problems and things like that. And then we collect a blood sample so we can look at genetics and immunology as well. And so this whole cohort has come back to us over and over again, so that this is a longitudinal study so that we can look at the development of their brain along with all of these other characteristics across childhood. So our first 3 time points are heavily weighted during early childhood. They come in once a year up until around age 6, and we've collected 3 MRI scans by then. And now we're just embarking on a middle childhood school age assessment in our cohort of girls, where we'll be looking at 9 to 12-year-old girls. Here we're going to have a focus on looking at the emergence of ADHD behaviors in girls, because this is something that is really, can be really disruptive for a lot of girls, and we need to understand better what ADHD looks like in autistic girls as it's emerging. And then the whole APP cohort, we are actually following them out into adolescence and early adulthood as well. So there's the boys in the original Autism Phenome Project cohort are now undergoing their 5th time point at 15 to 19 years of age. So it's a really unique study in that we get to follow this wonderful cohort of families across time and see how everybody is growing and developing and changing. So our current sample, just a few things I'd like to point out. We have a significant representation of autistic females. We are inclusive of individuals across the range of ADOS calibrated severity scores. So that's the diagnostic measure that is getting a score for autistic traits. And we have a range that goes across all of the scale, so 4 to 10. And I think really importantly, we have been working really hard to make sure we're representative and having plenty of children with intellectual disability, autistic children with intellectual disability in our cohort. So particularly in MRI studies, autistic children with intellectual disability have not historically been included in these studies. And of course, we know that of all individuals with autism, about 40 to 50% of individuals have lower IQ scores. And so as researchers, we need to make sure we're evaluating the entire range of people with autism, not just those with normal IQs. So in our study, we have 30% of our cohort are children who have IQs less than 70, another 17% in the moderate range. So pretty representative of the numbers out there. And then these are just the numbers that we have so far. This is probably a little bit outdated, 442 children at the first time point, and then all of the time points are still collecting data on, so these numbers are always changing. So what have we done with this data? So we have published a number of papers coming out of this cohort. I just wanted to focus on a few today that have looked at issues of differences between autistic males and females. And this is the study that I was mentioning before, where we wanted to look for a subgroup of children who had co-occurring psychiatric symptoms, even as young as age 3. So right when they were enrolled, we wanted to look for co-occurring psychiatric symptoms and then investigations with biological sex. We know from prior research that about 70% of older individuals with autism have a co-occurring psychiatric condition like anxiety or depression. We know that female adolescents and adults with autism have higher rates of mood disorders and internalizing problems than males. But what we hadn't yet looked at in the literature is what about in younger children? When do these symptoms emerge? Are there sex differences in the presentation of these symptoms? And then we wanted to also look at underlying neural differences. And so in this study, we focused on the region of the brain called the amygdala. The amygdala is outlined right here. It's in the medial temporal lobes. It's a very well-studied region in both autism as well as in the general population. We know that the amygdala is involved in emotion regulation and threat detection. It's been implicated in autism as well as other psychiatric conditions like anxiety. We know that the amygdala grows differently actually in boys and girls with typical development. So actually in many areas of the brain, there are slightly different trajectories of growth in different brain regions, such that girls will often reach the peak size of these structures about a year to a year and a half earlier than boys. And so we really need to think about when we are looking for sex differences, we have to really remember that there could also be sex differences in non-autistic individuals as well. And what we wanted to, our hypothesis in this study was to look, does this subgroup, if we identify, which we do, identify a subgroup of children with co-occurring psychiatric symptoms, do they have a different pattern of amygdala development as well? And so how did we do this? We used a data-driven approach to identify subgroups. We had 300 autistic children participating in this study, again at age 3. Parents rated different problems. So this is using the child behavior checklist. And in that measure, we can look at affective problems, anxiety problems, ADHD, as well as oppositional defiant problems. So anxiety and affective problems are the more internalizing types of problems, and ADHD and ODD are more externalizing types of behaviors. And I just list here some of the examples of the types of questions that then get coded into these different domains of psychiatric symptoms. These were our main, this was, I wanted to identify subgroups based on these behaviors, but we also put into our models adaptive functioning, cognitive ability, and autism characteristics. And so we entered the scores, all of those, into our subgrouping model, and then let that basically tell us how many different subgroups, subtypes that were identified in this autistic sample of 300 children. We wanted to know specifically, is there a subgroup with higher psychiatric symptoms? What's the proportion of girls and boys in this subgroup? Is there a different pattern of amygdala enlargement in this subgroup? And lo and behold, we did find different subgroups. We identified 3 different subgroups. A red group that comprised about 27% of the sample had high psychiatric symptoms, so that's here. They had high levels of depression, anxiety, ADHD, and ODD. They had relatively intermediate adaptive functioning, and this is measured by the Vineland. They had IQ scores that were very in the middle of the cohort, and autism characters. So this is the ADOS calibrated severity score that were slightly lower than the rest of the cohort. So that's about 27%. We had a purple group. This was actually the most kids in our sample who had 40% of the kids in our sample had low psychiatric symptoms, very good adaptive functioning, very good IQs, and lower autism characteristics. And then we had this green group of about 30% of our kids in the cohort who also had low psychiatric problems, but they had higher impairments, meaning they had lower adaptive functioning ability, lower IQ scores, and higher ADOS severity scores. So these were the three groups that we identified. And then we took a look at how it broke down across gender. And what we found was that 40% of the autistic females in our group were classified into this high psychiatric symptom subgroup, compared to only 22% of boys. So girls were overrepresented in this subgroup with high psychiatric symptoms. In contrast, there were relatively fewer girls in this purple group that had low psychiatric symptoms and lower impairments than the other group. There were only 30% of the girls in this group compared to 45% of boys. So we did find sex differences in these different subgroups, with girls being more diagnosed or more put into the subgroup with high psychiatric symptoms. What we found is when we looked at the amygdala was that we only saw amygdala enlargement in the high psychiatric symptom subgroup. So we didn't see amygdala enlargement in the subgroup of kids who had low psychiatric symptoms, suggesting that perhaps the amygdala is really more associated with psychiatric problems in this cohort of children as opposed to autistic behaviors. And what we found also was that the amygdala, the size of the amygdala, was associated with internalizing behaviors in girls. So the higher the score on those affective and anxiety problems in girls, the bigger their amygdala, but we did not see this relationship in boys. And so there seemed to be a sex difference in what the amygdala was doing in terms of these psychiatric symptoms across these boys and girls. Now, we also followed, because this was all at age 3, we also followed this subgroup out 2 years and looked at them at age 6. So we took the children who were classified into the subgroups at age 3 and simply asked, what do they look like now at age 6? What we found was that in the high psychiatric symptoms subgroup, the girls had, if they had high internalizing scores at age 3, they tended to also have high internalizing problems at age 6, but that was not the case in boys. In boys, their scores that they had at age 3 didn't really relate, weren't associated with the scores they had at age 6, so they were not as stable. Even though they were classified in high psychiatric symptoms at age 3, the boys then were not still maintaining those high psychiatric symptoms at age 6, whereas the girls were. And that's depicted here. This is now just showing the anxiety subscale, showing that this group with the high psychiatric symptoms, the girls tended to stay elevated, whereas the boys decreased to what the other subgroups looked like by age 6. And so there really could be something to this subgrouping early on, and that they're more stable in girls as opposed to boys. I know I'm running out of time here, so I'm just going to zip through these take-home messages. Even at age 3, we had a higher proportion of girls diagnosed with autism exhibiting clinically significant behaviors consistent with anxiety, ADHD, affective problems. Amygdala volume was more closely associated with these internalizing symptoms in girls than in boys. And our longitudinal follow-up at age 6 suggested these symptoms are more stable in girls, but not boys. So the sex differences are becoming even more apparent as the children are aging. And I think this all has implications for early detection, better understanding of neural mechanisms, so that we can lead to better interventions and treatments to improve outcomes later in childhood for these children. Okay, Very quickly, I'm going to talk about a work that was recently accepted and is in Press at Journal of Neuroscience. We looked at, in the previous study I told you about, we looked just at the amygdala. Now we're looking at the network of structures that talk to the amygdala in the brain. And what we are found, and I'm not going to go through this because I know we're running out of time and I want to leave time for questions. We looked at all of the regions in the brain that have monosynaptic connections with the amygdala. So this is a list of regions here. These are regions that have a direct white matter connection with the amygdala. And we looked at this as a network. We called it a network of amygdala connected regions. And then we looked for similarity and dissimilarity between each of these amygdala profiles. We call them across childhood. And the take home message is that we found sex differences. And so we found a different pattern of regions contributing. So these are different regions that were sort of showing up in males. The middle temporal gyrus, the anterior cingulate cortex were showing up as more different in males, whereas in females, we were seeing bigger differences in the fusiform gyrus, the entorhinal cortices. And again, these are regions that have direct connection to the amygdala. They both show differences in autism, but they're different regions that are being impacted. I'm going to skip through this slide because it's a little bit complicated, and this one does not actually have a sex difference component to it. But I think it's important to talk about this study just briefly because we're going beyond just looking at one single brain structure, the amygdala. We're looking at a network of regions now in terms of the size of these regions. And there are regional patterns that are differentiated across sex. So girls had bigger differences in the development of the fusiform gyrus, which is involved in face processing, and the entorhinal cortex, which is highly involved in memory processes, whereas boys differed more in the anterior cingulate cortex and the middle temporal gyrus, which have been more implicated in prior autism studies, probably because those studies were done primarily in boys. That last slide that I skipped, the take-home message of that was children with greater social impairments at age 3 showed the largest divergence in their amygdala-connected regions across childhood. And so we have looked for other sex differences across other parts of the brain. I won't go into any of the details here, just to say that we've looked at the corpus callosum, which connects the two hemispheres of the brain. We've also done functional studies of the amygdala. We've looked at overall brain growth and found sex differences in all of those areas within our cohort. So I'm just going to wrap up just to say that the study I see that we've done so far is really just the tip of the iceberg. But I think all of the studies that I talked about, in addition to our studies on brain development, we need to focus on earlier recognition and diagnosis of autism in females. It's helpful to have large-scale multidisciplinary studies, critical to have sex-balanced samples and representation across the entire autism spectrum so that we can really truly understand both similarities and differences across autistic males and females. Longitudinal studies, I think, are really important because things change over time. We're really interested now in looking at the effects of puberty on brain development as well as behavioral development now that our cohort is reaching that age. And then finally, we really need to identify, evaluate gender identity and gender diverse and non-binary identities layered on top of all of these sex differences that I'm talking about here. Finally, I just want to mention, I know there's a lot of practitioners and perhaps some families out there. This NeuroTeens group is a virtual social support group that my lab started about a year ago now. And we started this based on one of our research families from the GAIN study asked me if I knew of any social support groups specifically for autistic girls, because her daughter was in high school and was struggling, but all of the autistic support groups that she joined were male dominated, and she just wasn't interested in socializing with them. And we looked around, we didn't see any appropriate groups for her to join. And so we started one. Initially, we were thinking about having a group specifically for girls, but we wanted to open it up to non-binary teens as well, because they're another underserved group. We meet weekly. This is a group that's for 12 to 18-year-olds. It's not a clinical group, but it's just a place where they can meet each other, support each other, understand that they're not alone, and just let teenagers be teens. So with that, we are going to be starting another session on our NeuroTeens. It is virtual, so people can join from across the country. And so if you're interested in learning more about that, you can send me an email and I can give you more information. So I think that's it for my presentation. I just want to acknowledge all of the people who've been working on the study over the years. I'm collecting all of these MRI scans. These are pictures from the Mind Institute thank you party that we have each year for research participants. We have a lot of fun with that. And then our funding acknowledgements. Some of this work was actually funded by the AIRP. Josh Lee was funded by this to do some of that amygdala network work that I talked about in that last slide. So I'm very grateful for that funding as well. And so with that, I will stop and take any questions. And I know Brianna wanted to talk about next things coming up.
Oh yeah, what a wonderful presentation. Thank you so much for that and your work in this area. So we do have some time for questions. We can leave up that slide. That's just our feedback survey if anybody has time to take that. Otherwise, I'm going to start with some questions that we have in the chat. This was actually pertaining back to slide 9, so feel free to jump back if you want. Somebody asked if you can just say a little bit more about the CNVs.
Oh, yeah, sure. Sorry, let me go back to that. I'm not going to say a whole lot more because I don't, it's not my area of research, but I did just sort of gloss over what copy number variants are, if that's what you're asking. Basically, they're duplications or deletions in the this is a slide just depicting a copy number variant and what this specific type of genetic mutation is. And there have been, I think, 2 studies now showing that autistic girls who are diagnosed with autism have higher frequencies of these copy number variant events. And the copy number variants that they have also tend to encompass a larger section of the gene. So it's sort of both sorts of things. It's really just suggesting that there are different genetic pathways to get to autism. And it seems that females have more frequent and more extensive genetic hits than do the boys. Did that answer your question, Caroline? Hopefully. I'm sorry, I'm not a geneticist, so that's really all I can say about it.
And then, it was answered after that. We have from slide 14, does the behavioral social splitting continue to exist in adulthood and even in later life for autistic females? What are the implications for social isolation and how can society, services, people mitigate the negative outcomes?
So Laura, that's a great question. I don't think that there have been specific studies on social flitting in adults, but I would absolutely think that that is still happening in adults as well. And I think social isolation is a big problem, leading to higher rates of depression and anxiety in autistic women, definitely leading to negative outcomes. And so, NeuroTeens is a small example of what we're trying to do to try to help with this social isolation that autistic females may be feeling in non-binary teens. Just trying, and it's not been terribly hard to just bring together people in a virtual room that maybe have some commonalities, and it really does help alleviate some of that social isolation. I'm not saying that it's solving everything, but I think the more we can have groups like this, the better it will be. How might our results inform future interventions? Primary care. I think, one thing that we're really trying to tackle is just raising awareness that autistic girls exist and need to be diagnosed and maybe need to be probed a little bit more to get an accurate diagnosis, I think primary care is a great place to start. I personally have friends in my lives who have girls who are 9 and 10 years old before they finally get a diagnosis of autism. And what they tell me is, well, my primary care physician said, no, it's not autism. Girls don't get autism. I think we really need to change that and just make sure we're, that's why I give presentations like this. The more people that can know about what's happening in terms of misdiagnosing or underdiagnosing girls, the better. I know a group of us, autistic researchers, or no, sorry, I'm not an autistic researcher, researchers focused on autism and specifically on girls. We're trying to write more guidances for primary care physicians in terms of how to identify girls and how to identify their unique needs and challenges, and making them more aware. So that's really what we're trying to do. And then I see one more question here. I can just address that, Brianna. Neurodiverse groups for girls over 20. We do in Sacramento, I know that we have a women on the spectrum group. I don't know where you are, Camilla, if there are any groups like that. My hope is that there will become more specific groups for individuals. You can reach out to me. We do take girls up to 18 in our group. But of course, 18 is just a number in terms of age appropriate and developmental level. It could be different. So if you want to reach out to me, Camilla, I'd be happy to talk to you more about our neuroteens group and if it may be appropriate, or our group in Sacramento, if you're from this area.
And then we got another question in the chat here. Any tips for practicing clinicians in screening or identifying women and girls?
Especially those that require low support. So, so I'm not a clinician, but the clinicians that I've talked to at the Mind Institute who do the diagnostic assessments for us, what they're focused on is, when you're administering an ADOS or you're doing an assessment, you just have to try to continue probing different sorts of scenarios and behaviors, maybe spend a little bit more time doing the ADOS and interviewing the child, just to make sure that they are, that you're getting an accurate picture of their full diagnosis. Getting the parents' input as well, I think, is really important, and really believing the parents, because they're with the child much more, and they can see them asking. You really can't tell, like Dr. Bishop in that one assessment. And I've seen girls come in for their first diagnosis, and I always think our clinicians are magical because, they can come in and they can look fine, non-autistic to me, and then when they start doing the ADOS with them, they're just really, going at trying to pull out these social challenges and probes in multiple ways over and over to try to draw out these behaviors so that they can see them better. I know that's not a great answer, but there is guidance coming out in terms of, clinical care and things like that. And I know that a group of us are working on a commentary right now, and hopefully that will be out soon. That does have a lot more practical guidance for clinicians. I can keep you posted. I'll just say, if I can just say, I'm a neuroscientist. I got into this field because I was interested in the brain. I'm thrilled to be talking to a group like this and starting something like Neuroteens, because I've sort of recognized that just studying the brain is not going to be enough to help this community of individuals. And that's why we've started things like Neuroteens. That's why what we're trying to do more and more is raise awareness and educate the people, the clinicians who are actually treating and working with these families, just to answer any questions. And so I would just throw it out to anybody out there. I'm always happy to come speak to parent groups or things like that, that just want more information about girls and autistic women. So just throwing that out there.
Great. Thank you again. And either you can stop sharing and I can share. I'm just going to go back over the next webinar, and sorry about that. Okay. And sorry, my screen, I'm on two screens here. So basically just the next the survey, and then this is for registration for our next webinar, will be by Dr. Dora Raymaker. She's going to be talking about system approaches in autism services research, especially pertaining to holistic research or holistic services. So I will also place the link to register in the chat. Other than that, feel free to follow our social medias. Thank you all for attending. Thank you, Dr. Nordahl, again for a wonderful presentation. Hope everyone has a wonderful day and take care.